Good morning children, I am Isis. Now I am going to teach you T.H. Morgan's experiment on Drosophila. In the last class I taught you about chromosomal theory of inheritance by Walter Sutton and Theodore Boveri, and the experimental verification of their chromosomal theory of inheritance was given by T.H. Morgan, who was conducted an experiment on Drosophila melanogaster or fruit fly. Let's see. Okay. So T.H. Morgan, Thomas Hunt Morgan, and he was conducting an experiment on, the experimental material was Drosophila melanogaster. Drosophila melanogaster. It is the fruit fly. Okay. And he was conducted an experiment, monohybrid and dihybrid crosses in this particular material of Drosophila. And in his experiment, he was given experimental verification for chromosomal theory of inheritance, the basis of variation and linkage also. So he was given the experimental verification for chromosomal theory of inheritance and variation. He was given experimental verification for variation and he was given experimental verification for linkage also. So what is linkage? We can see now. Linkage means the genes present in the chromosomes are linked genes. A pair of genes are there in the chromosome, and some linked genes remains linked during inheritance also. That means the phenomenon of certain genes staying together during inheritance, and that phenomenon is called linkage. For example, in the last class we learned about the round yellow. So the experiment conducted with a round yellow and wrinkled green. Here capital R, capital Y is the genotype, small r, small y is the genotype of wrinkled green. In the F1 progeny, we got round yellow, and in the F2 progeny, we are having the round yellow with the same genotype. Am I right? And we are having the wrinkled green with the same genotype. That means these linked genes remain linked in the offspring or in the next generation also. And that property of this linked gene which remain together or which remain linked in the offspring also, and that phenomenon of the gene is called linkage. Is it clear? So the linkage is the phenomenon of certain genes which stay together during inheritance also, or we can say in the next generation also they are staying together, and that property of gene is called linkage. At the same time, we are having some offsprings with the this genotype, that is round green. Am I right? That means a combination of two parents. So the gene which are reshuffling here to form a new combination. So this is an entirely different from the parental characters. Am I right? Or we can say there is a combination of the parental characters, and that is called recombination. This property is called recombination. Is it clear? So the linked genes can show recombination due to crossing over. There is a crossing over occur here. Is it clear? So linkage is the phenomenon of certain genes staying together or remains linked in the next generation also, or during inheritance also. And that property is called a linkage. And recombination means the genes which are reshuffled to form a new combination in the offspring, and that is called recombination. So this gene is recombinant genes. Is it clear about linkage and recombination? So T.H. Morgan, to his experiment, he was given experimental verification for linkage and crossing over or recombination. Is it clear? Now we can see why he was selected this Drosophila melanogaster or fruit fly as the experimental material. What are the reasons for selecting experimental material Drosophila? The reason, first one, it can be grown on simple synthetic medium in the laboratory. That is the first reason. And second reason, it completes its life cycle in about 2 weeks. So within a short period of time, you can generate many offspring, or many generations can be raised within a short period of time, and produces numerous progeny flies, and can breed. They can breed throughout the year. And next point is, there is a clear differentiation of sexes. That is, the male and the female flies can be easily distinguishable. And next, many types of hereditary variations can be seen. And the last point is, it is very less expensive to rear. So these are the reasons for selecting Drosophila as the experimental material or experimental organism by T.H. Morgan, Thomas Hunt Morgan. Is it clear? Now we can see, I taught you, he was conducted monohybrid as well as dihybrid crosses on this Drosophila. And let's see a dihybrid cross made by T.H. Morgan on Drosophila. So this is Drosophila melanogaster or fruit fly. Okay. So what he has done actually, he was done 2 crosses, 2 dihybrid crosses. In cross A, he was selected, the female was having yellow body with white eye, and it is crossed with the male. Male is having brown body with the red eye. This is the male. And this is the mutant type. And this is the wild type. Okay, I'll show you the Drosophila. Here you can see. So this is the female one, yellow body, white eye, and this is the male, that is wild type, brown body with the red eye. So he was hybridized the yellow-bodied, white-eyed female with the brown-bodied, red-eyed male. And the female is the mutant type and the male is the wild type. Okay. Now we can see the result. Here, so this is female and this is male. And in the first offspring or F1 generation, he got the offsprings with both the parental characters. And the parental characters such as yellow-bodied, white-eyed offsprings are male. So in the offsprings, the male are showing the female parental characters. This is his observation. Okay. And the female offsprings are showing the male parental characters such as brown bodied, red eyed. Clear? So in the F1 generation, the offsprings are showing parental character. So anyway, male is showing female parent character and female is showing male parental character. And now in the F2 generation, along with this parental character, so this parental character, yellow body, this is mutant character, am I right? So we can say in the offspring the male show mutant character and the female show wild character. And in the F2 generation, the offsprings are showing parental characters such as mutant and wild type. That is the parental character. Parental genes or parental character, that is 98.7% of the offsprings. Sorry, 98.7% of the offsprings are showing parental characters such as mutant and wild character. Which are the mutant and wild character? Yellow body, white-eyed and brown body, red-eyed characters are shown by the F2 generation in 98.7%. And along with the parental character, some recombination or recombinant character. Recombinant character are also shown by the offspring 1.3%. Is it clear? So let's see which are the recombinant character. Here the parental characters you can see mutant and wild character. Along with the mutant and wild character, the F2 offspring show yellow bodied, red-eyed and brown bodied, white-eyed. So this is the recombination of the parental character. Is it clear? The parental characters are yellow body, white-eyed and brown body, red-eyed. And that was shown by the F2 generation in 98.7%. And along with the parental character, the flies shows a recombination, a combination of both the parental characters such as yellow body, red-eyed and brown body, white-eyed. And that is only 1.3%. Is it clear? So this is the result of cross A. Next we can see another cross, that is cross B. Okay, so keep this percentage in your mind. Next we can see cross B. In cross B, he was conducted the same dihybrid cross with the female is yellow bodied miniature wing. Miniature wing. And that is the female. Okay. And it is crossed with the white bodied normal winged male. Is it clear? And now the same happened here also. The same offsprings are there. In the F1 generation, they are showing both the parental characters. Which are the parental characters? Yellow bodied miniature wing and the white bodied normal wings were shown by the F1 offspring. At the same time, in the F2 generation, along with the parental characters are there in the percentage of 62.8, and there is a recombination of the parental characters or a combination of both the parents in 37.2%. And what was the recombinant character? Yellow body, normal wing. Am I right? That's right. Okay, you can write over here. So the chance of recombination is yellow body, normal way. Normal way. And another chance of recombination is white bodied, miniature way. Miniature way. So, this is the observation done by Morgan after conducting experiment dihybrid cross in Drosophila. So, what was his conclusion? You can see the parental characters are more than the recombinant character. Am I right? So, this is the conclusion of Morgan after conducting experiment in Drosophila. Let's see Morgan's conclusion. And Morgan's conclusion was, when two genes in a dihybrid cross are present in the same chromosome, the parental combination, the proportion of parental combination is much higher than that of the proportion of recombination of gene. Is it clear? So in the first cross, we can see cross A. We got parental characters or parental characters showing offsprings in 98.7% and recombinant character 1.3%. And in cross B also, we can see the parental characters 62.8% and recombination 37.2%. So from this he concluded that when two genes are present in the same chromosome in a dihybrid cross, the proportion of the parental characters are higher than that of recombinant characters or recombinant genes. The proportion of the parental genes are more higher than the recombinant genes. That was the conclusion. And the genes which are tightly packed or tightly linked show only low recombination. Only low recombination, and the genes which are loosely linked show higher recombination. High recombination. Is it clear? So this was the conclusion of Morgan. And these genes on chromosome, I taught you genes are located on chromosome, and these linked genes are linearly arranged in the chromosome. And these linked genes always staying together during inheritance, and that phenomenon is called a linkage. And these linked genes show independent assortment due to crossing over of homologous chromosomes. If there is a crossing over, these linked genes show recombination. A graphical representation of this linear arrangement of genes is called chromosome map. So the chromosome mapping was constructed by Alfred et Sturt-Event. And the chromosome mapping is the linear graphical representation of the sequence of genes, and the relative distance of various genes in the chromosome also can be represented as a map, and that is called chromosome map or chromosome mapping, and that phenomenon is the mapping. Let's see. Chromosome mapping. Chromosome mapping is a linear graphical representation of the sequence and relative distances of the various genes present in the chromosome, and it was constructed by Alfred et Sturt-Event. Is it clear about Morgan's experiment on Drosophila and chromosome mapping? That's all for today.